synthesis has two major steps. The first is transcription, followed by translation. Transcription is when the DNA is opened up by an enzyme called DNA helicase. And once it's opened up, an enzyme called RNA polymerase will come in, move down the exposed coding section of the DNA, and create a transcript known as messenger RNA. So it basically copies the coding sequence, also known as a gene, and then that in turn is transferred out to the next step, which is called transcription. Transcription is when the messenger RNA, which was transcribed from the DNA, is brought out through the nucleus and into the cytoplasm where it encounters the ribosome. In this instance, our transcript, our messenger RNA, is UAC, GGG, AAC, ACU. That must mean that the original DNA was A, T, G, C, 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 T, T, G, T, G, A. I'm reverse transcribing it just to show you how I got there. So once this is encountered by the ribosome, it's getting ready to go to work. But first we need some raw materials to build the protein from. So in this instance, this cell is taking place in a dog. This same process occurs in all living things that make protein. So I could pick a dog, an aardvark, a hippopotamus, or a tuna fish, or a human. And everything we're talking about here is the same. This dog right here is eating a wiener, a hot dog. It's taken to the stomach, it's broken into its constituent parts. A protein would be broken down into the monomer, amino acid. That is absorbed by the bloodstream, pumped to the cell, and the hot dog, which is actually a polymer of triangles, squares, and circular amino acids, is broken apart and carried in as circles, triangles, and squares into the cell. These are encountered by a different type of RNA called transfer RNA, also abbreviated as tRNA. tRNA is extremely specific. Each tRNA is identified by a sequence of 3 base pairs called an anticodon. And this anticodon will only pick up a particular amino acid. We don't have these named up here right now. We have instead designated them by shape. So CCC picks up only circles, AUG only picks up triangles, and UUG only picks up squares. Now, we've loaded the amino acids onto a particular transfer RNA, while the ribosome, also known as ribosomal or rRNA, is beginning to read the code. Immediately, it looks for a transfer RNA that has a complementary sequence to the UAC, which would be AUG. And we have one right here. So it navigates this into position, causes it to lock into place with its load. Then it reads the next sequence, GGG, so it's looking for an anticodon to match this codon of CCC, and here it is right here, the circle. So it's brought into position, it bonds into place. As soon as it bonds into place, the circle bonds to the triangle, and the triangle's transfer RNA pulls away, looks for another triangle if the dog continues to eat wieners. Meanwhile, the next sequence, which is AAC, is encountered by the ribosome, and it commands another transfer RNA with the code UUG to come in, and here's one here. So it navigates the square into place, it locks down, and the instant it locks down and bonds to the circle, the transfer RNA for the circle pulls off, and it goes to find another circle. And then the ribosome reads the last code here, which is stop, so it immediately liberates the transfer RNA carrying in the square. It goes off, and now we have a free-floating polypeptide, called a polypeptide because it's poly, many, peptide, bonds. Those are the type of bonds that hold them together. This is then carried to the endoplasmic reticulum or some other location in the cell where it is folded into the correct shape and becomes a functional protein. That's it.
Okay.